Good morning. My name is Rajesh Naik. I am from the Air Force Research Lab in Dayton, Ohio, and I'm here to talk to you about some work that we've been doing on how we exploit biology to build materials. I'm a biologist by training, and when I first joined the DoD, I was wondering, especially the materials directorate, how am I going to use soft, squishy materials to address some of the extreme challenges that we face in the aerospace arena. And what I'm going to talk to you about is some of the basic research work we started with and some of the off-ramps that it created. We ended up in a place completely different from where we started, and I want to share with you some examples of what we've done over the past couple of years. So if you look at nature, humans for centuries have been trying to exploit some of the things that you see when you go into the aquarium, so you go for a walk in the park, some of the exquisite and phenomenal structures that you see from biological systems. For some reason, this is not working. All right, got it. Some of the phenomenal structures that you see in biology, right? For centuries, human has tried to emulate some of these properties, and with now the advancements in biological sciences, you're getting a better understanding of some of those molecular devices, some of those nano devices, machines that are created by nature, but all the way to high order structures like how do marine organisms camouflage in the environment. Okay. Using imaging tools, using molecular biology tools, we're beginning to get a better understanding how the structures are formed. How do they self-assemble nanoscale devices and how you could use them. How can you understand some of the structural property relationships in some of the materials that are made by nature, and how can you exploit some of those processes for DoD applications? And we and others have been using some of these approaches. For example, understanding how insects sense pheromones. Can you create better sensors that are much more sensitive and selective against different targets of interest? Can you understand how does biological systems control reactions and create better catalysts, if you can use them to cap or control nanoparticle sizes, for example? Can you create functional materials? It could be a hybrid of both a bio component and a non-bio component, but can you create hybrid materials that have enhanced properties? And also decontamination materials. Can you take principles in biology to create materials that can decontaminate against chem bio agents? So one of the class of functional materials that we were interested, the Air Force is interested, is in energetic materials. Now, energetic materials have applications both in the military and in the civilian space. In the military, in terms of munitions and propellants, but also for micro propulsion systems. And for example, in the civilian sector, you use them to make airbags. Okay. And they're all based on the energy density of materials, and they come in different classes, right? The most common one everyone knows of is fuels, liquid fuels that you use to gas up your car. Right? They have low energy densities. You have small molecules, things like TNT, RDX, CL20 that the DoD has been involved in developing. To metal nanoparticles or metals, they're highly energy dense, but is at the smaller size scales that they are highly reactive. And then you have the food groups too, right? Like a burger, right? They're very energy dense. It's also an energetic material. Just be careful how much you eat of those things. You don't want to explode. So again, as I mentioned, the class of materials we were interested in is energetic materials. Now, energetic materials have their own issues, right? The class of energetic materials that we were interested in were called thermites. Thermites is when you combine a metal, which is a fuel, in this case could be aluminum oxide, with an oxidizer, and they could be things like perchlorates or ferrites or metal oxides. You mix them together to create a binary complex, and that's your energetic material. You ignite that and it goes boom, right? But there's some challenges with that. The mass diffusion issues, the heterogeneity issues that comes with it. So as I depicted in my graph there in yellow and blue colors are the fuel and oxidizer components. And if you look at real life, when you go and synthesize nanoparticles, they come in all different sizes and shapes. Even if you have a nice control, you still have issues with controlling the size of the nanoparticles, in this case nano-aluminum. And the rest of my talk is going to focus on the use of nano-aluminum as the fuel source. So what you do is you mix those two, try to get them in the right stoichiometric ratios to get the maximum bang for your buck. But there are challenges. Combustion kinetics are limited by mass transport, diffusion distances, and other things. So that's why people have been trying to go into the nanoscale, shrink everything to small size, and get very high concentrations of both the oxidizer and the fuel. But when you do that, you also create other issues because they become highly unstable, not easy to transport, so on and so forth. So if you look at nature, nature has a wide range of redox materials. And we became interested in one called ferritin. Everybody has it. We have it. So you don't get iron poisoning. There's a structure that sequesters the free iron in the bloodstream and encapsulates that into a proteinaceous structure called ferritin. And within the core of that ferritin is an iron oxide particle. Lots of people, including my group, has used this ferritin cage as a way to insert other things, including oxidizers into those structures, and it protects them with that proteinaceous core. So when we're doing these experiments and said, okay, let's use the iron oxide from nature, mix it with aluminum oxide, create now in the same size scale, because the size of this is about 12 nanometers. Some of the nanoparticles that we were using for nanoaluminum for the energetic materials were around 100 to 50 to 100 nanometers. So they're within the same, an order of the same size scale. So we mix those things together. We got combustion reactions, but we were not very impressed. Okay, we were not going beyond the state of the art. So we came up with a new technique where what we call the formation of nanoliquids or ionic liquids, where you can take proteinaceous molecules, functionalize the surface using traditional chemistries, so you can increase the concentration of iron oxide. Okay, so when you go buy milk, it's in a solution phase with colloids in it. If you try to boil the milk, what happens? It denatures, right, and then just precipitates out in solution. Okay, no more flows like a liquid. Right? In this case, we can get to very high concentrations by doing functionalization chemistry. So I showed with that glow around those particles, and we can create an ionic liquid or a protein liquid. And those things flow like a liquid, but has less than 3% water in there. Okay, so very high concentrations of the iron oxide particle, and then what I can do is I can now add my nanoparticles and get a nice mix without the presence of water in there, and now I can get very high concentrations both of my oxidizer and my fuel. So I can add about 25% weight volume into that mixture and increase both the concentrations, trying to get to the stoichiometric ratios of both the oxidizer and the fuel, so you can get the maximum efficiencies from your energetic material. Okay. So we tested it, and you can see this is just standard combustion reactions that you can measure. You can take high-speed video to look at the characteristics of the combustion reactions, capture the flame profile, so on and so forth. I am not an expert in combustion materials, but working with collaborators, they've been able to help us understand what's happening in there. What's the take-home message here is you can see the difference between conventional material and using our ionic liquid-based proteinaceous material when you mix them. You get a very high energy output, exothermic reaction that comes out of it that almost reaches the theoretical limit of what you can get out from that system. But what was more interesting is I was just saying these things flow like a liquid, but there's no water in there. So you can use things like 3D printing. You can cast them into molds. You can do all sorts of things to it. So this is just printing those things onto substrates, onto electronic devices, so on and so forth. So if you want to have anti-tamper devices, you can use this approach where you can now use, if someone opens a package, use light or something that will ignite the reaction, and you get a combustion reaction to kickstart. We can make crayons that you can write with if you want with this, not for kids, but you could write with this structure. Again, using just very simple processes that we developed in the lab. A picture is worth 1,000 words, but a movie is even better, right? So what I want to show you is movies of combustion reactions where you can see this is slowed down because the reactions are very fast, comparing conventional materials to a bio-derived material. And what you're going to see the difference between the two. Okay, this is your bio-based material, and this is your conventional material. You can see the exothermic reaction that you get from the bio-based material, and the combustion materials is just still taking off. So this is a couple of experiments we did over the last couple of years that have been published. But during the course of these experiments, we found out that when we were creating these ionic liquid-based proteins, we found that the proteins became highly stable. Right? They were not subject to denaturation when you kept them at room temperature on the bench top. So today you go to the grocery store, you buy eggs, you buy milk, You have to put it in the fridge. If you keep it out for extended periods of time, what happens? They get spoiled, they denature, things start to settle down, funky things start to grow in them, okay? So that's why you have to refrigerate molecules. So for example, antibodies and diagnostic reagents have very limited shelf lives. You have to refrigerate them, so things like vaccines and therapeutics are always kept in the cold. So if you go for a vaccine shot, they bring it out of the refrigerator and give you a shot, right? Because it's not stable at room temperature. We did the same thing with antibodies. We said, let's take antibodies and see if you can stabilize them. So we create ionic liquids of antibodies. They still flow like a liquid, but there's no water in there because water is one of the factors that enhances denaturation of biomolecules. And we heated it up to 100 degrees, okay, for an hour and said, will it survive? And this is just showing you an assay called an immunoassay. When it binds to an antigen, you can do a colorimetric-based approach to detect it. And you can see an ionic liquid-based antibody can even survive when you heat it up to 100 degrees. Whereas the standard one is pretty much has little or no activity. Okay. Why is this important? From a commercial standpoint, or on the civilian sector, think about vaccines and other things in resource-limited settings. It has a huge logistics chain that has to go with it if you want to administer this in the field. Not only in the civilian sector, in the military field, when we take care of wounded warriors in the battle space, things like vaccine and other therapeutics, you also have a cold chain logistics that have to go with it. It's very expensive, it's bulky, and you have to plan for those things. A lot of times, a lot of reagents gets wasted in the billions of dollars. We lose a lot of biological reagents due to the breakdown in the cold chain logistics. So can you use some of these approaches to stabilize antibodies and other things to maintain their activities at extreme temperatures? As I showed you, we could heat some of those antibodies to 100 degrees. We have kept them about 6 months at room temperature, and they don't degrade, okay? We might lose 10% of the activity, but they remain stable after extended periods of time. So think about this. Maybe you eliminate the need for refrigeration and cold storage. You have a powder, you just add water, and you're ready to go. And that is one of the technologies that we have developed. And we have a small company that has licensed this technology from the Air Force and using to develop diagnostic kits for the Red Cross. So next time you take a walk in the park or you go to the zoo with your kids, think about some of those things that biology does well and how can you use them to solve challenges that we see today across. Whether it is trying to understand how do plants develop some of those unique structures and how can you use some of those structural properties to develop better airframes, maybe better mechanical stability structures for other applications. It's a fascinating time to be a biologist in my mind. I'm a biologist, but with all the tools and that you see the boom that's occurring in the biological sciences, there are lots of application. And I think some of the basic research funding has enabled us to do that through the Air Force Office of Scientific Research that has funded this work. And I'd also like to thank a brilliant team that has worked with me on this project from the Air Force Research Labs, and I thank you for your attention.